Good evening. It's a real special pleasure for me to be here today to talk to you about some of the work that's been ongoing in my laboratory. As you know, I've spent a good part of my life as a clinical cytogeneticist, and I can tell you that there were many days along that path which were pretty disappointing in terms of delivering information for families that they could make a decision about a pregnancy based on, ranging from times early on in my career when we would tell them that this variant, such as an inverted duplicated 15, would range from having no phenotype to a very abnormal phenotype. So I think that we're really getting to a time now, and that's what I'm going to talk to you about today, where it's going to be much more satisfying in terms of being able to make better predictions about clinical phenotypes. So you may see that I have the logo of the University of Manchester also on my slide. Since July, I am enjoying an adjunct appointment there in their Department of Audiology. And I'm just going to sneak two slides in that have to do with my work on the genetics of hearing loss to tell you about a couple things that are going to start to happen at Brigham and Women's Hospital. So we have a project which will be funded by the NIH called Seekaboo for sequencing a baby for an optimal outcome. And our plan is to offer whole genome sequencing to trios when the baby has failed their newborn hearing screening test in the hospital. So every baby is screened first and within the first 48 hours of birth, and if they fail that first test, then they're screened again. And then if they fail that second test, they're referred to, in fact, the state of Massachusetts has a very high follow-up rate because before the baby leaves the hospital, the parents have already scheduled the appointment, the follow-up appointment with the otologist. So within 6 weeks or 8 weeks, the baby sees an otologist and has that hearing loss either confirmed or not. And at that point, at least in our state, about 20% of those cases are actually confirmed to be true hearing loss. So this is a little bit complicated schema to show you. We're going to be enrolling babies at the Brigham again, the newborns, and then they'll be followed up at either the Boston Children's Hospital otologist or Mass Eye and Ear, and we'll be doing surveys for parents who don't choose to enroll to find out what reasons they didn't want to undergo genomic testing. And we'll have a standard of care arm at the Mass Eye and Ear. And I'm really excited to do this because I think hearing loss is a terrific system to think about really validating that it's going to make a difference to do genomic sequencing and to have an early diagnosis in terms of management. And we'll also be looking at how the management of that baby is different, when they have a genomic diagnosis, when they go for that confirmation with the otologist. So stay tuned. I sort of hope that this might be something that we will also think about doing in Manchester, and we're beginning to have conversations there about it. So for today, I'm going to talk to you about a project that we fondly know as DGAP, for Developmental Genome Anatomy Project. And this is a project that's, the hypothesis is a paradigm in human genetics that chromosomal rearrangements in individuals with congenital anomalies can be etiologic in the abnormal phenotype due to disruption or dysregulation of genes that are critical in human development. And this really harkens back to the sort of mid-1980s when we really began doing positional cloning, using variants in chromosomes to tell us where to look for genes involved in that disorder. So the contributions that I'll talk to you about the project today are sort of divided into these three categories: gene discovery, gene annotation, genome annotation, and genomic structural rearrangements. And I will have to admit that when we conceived of this project in early 2000, I really was only thinking about gene discovery and genome annotation. I didn't know that we'd also be really learning some basic biology about genomic structural rearrangements. Had no idea that we would transition this into the clinic in terms of informing and making more accurate prenatal diagnoses. And then recently trying to develop a standardized nomenclature for human cytogenetics to move to reporting at the nucleotide level. And with regard to this, I know there's a lot of people who like to think that cytogenetics is just something of our past, but I was really inspired by the words of a wonderful cytogeneticist, Dorothy Warburton, who reminded us through sort of a blog that we have for cytogeneticists that cytogenetics is not a method. It's not G-banded chromosomes or fish. It's a discipline. And although the way we look at the chromosome may change, so instead of looking down the microscope, we'll look at sequence, that the problems remain the same, and we have to understand basically the structural rearrangements and the information that we need to get from them. So the project, sort of in overview, is obtaining a blood sample from a subject and then doing a next generation sequencing approach that we refer to as a jumping library, which I'll explain in just a few minutes a bit more, to identify the rearrangement breakpoints. Knowing the vicinity of that breakpoint reads over the breakpoint region, we then develop PCR primers and sequence to nucleotide resolution and identify candidate genes. Now, the project, as initially conceived and performed during the first sort of 5 to, I would say, maybe 8 years of the project, was to validate every candidate gene by either other human patients that had an overlapping phenotype or animal and cellular models. So although this was a fabulous developmental biology project, with the advent of next generation sequencing, we can get to the breakpoint so quickly that we're sort of not able to do that type of validation in a timely manner for providing this information back to clinicians and families. So although we still are very interested in knowing deeply how many of these genes cause the phenotypes that we see, probably that work is going to be done by lots of people and not just our group in terms of looking deeply at a particular gene. And now we are beginning to use what we call convergent genomic evidence, using a lot of database mining and understanding from that perspective to incriminate the gene that is a candidate from the chromosome rearrangement. Now, I'm a big fan of genome-wide association studies and have been involved in that in pretty deeply in uterine fibroids. And we're in the process right now of doing a GWAS for age-related hearing loss in the UK Biobank subjects. But in this particular project, and of course GWAS is really great because you don't have to have any particular hypothesis, but here we can actually look under the lamppost, because all the keys are under the lamppost. I can tell you that when there is an abnormal phenotype and there is a balanced rearrangement, there's a really good chance, a de novo balanced rearrangement, that that chromosome rearrangement is involved in the etiology of the disorder. So to date, we've enrolled over 300 families, have sequenced over 150 cases, and identified over 200 genes. Most of those rearrangements are simple in that there's just an exchange between two chromosomes, or there's an inversion within a chromosome. But one thing we learned was that many of these rearrangements, which look simple at the level of the microscope, are not simple. And that led us to an understanding of germline chromothripsis, where a simple rearrangement might have 14 breakpoints localized in that particular area, or you may even have chromosomes involved in the rearrangement that you didn't suspect. So one of our favorite cases was a presumably balanced 3-5 translocation. It turned out there wasn't a single junction fragment between a portion of chromosome 3 and a portion of chromosome 5, because there was also the involvement of chromosome 7. So that number has stayed around 20%. So about 20% of these apparently balanced rearrangements are actually quite complex. And then we have others that we have not yet solved. So this was the gene map. And you can see during the first part of the project where we sort of were doing fish mapping and southern blotting and cloning the rearranged fragments. We had put 19 genes on the map, and those are indicated in blue. And then you can see what happened with next generation sequencing, just an explosion of gene discovery from these chromosome rearrangements. And then some in green that were from the time period after we had submitted our grant for renewal. So I told you that we use a library method that we call jumping libraries, and it's essentially similar to what you think of in terms of next generation sequencing. The only exception is that we're cloning a bit larger fragments, so 2 to 3 kb fragments, so that we'll span the breakpoint region. And I just indicated, let's see if the pointer here, that sort of a hypothetical rearrangement between chromosome A and chromosome B. Of course, this could be an inversion, which would be the same chromosome, but we're looking for those fragments that don't align to the reference sequence. And we know what we're looking for because we're in this project, because we've had all the patients are referred because they have a chromosome translocation or inversion and a phenotype. And then again, what we do is develop primers from knowing where we are and then sequence the breakpoint to nucleotide resolution. So I'm going to start with the gene discovery and sort of transition you to what I think is a really exciting time now for us, especially clinical cytogeneticists. So this is one of my favorite cases, known as DGAP 103. This was a young boy who was referred to us when he was about 8 years old, who had clinical features that are pretty remarkable for an 8-year-old. Multiple lipomas, very benign fat tumors. Really impressive overgrowth. Actually, his height, weight, and head circumference were greater than the 95th percentile. He had arthritis, advanced dental and bone ages, bilateral bowing of the legs, and brachydactyly of the hands and feet. So just think a minute, why this progressive or impressive long bone overgrowth, but then the brachydactyly? And that's part of the whole story here. That's really become very interesting. And facial dysmorphism. So here's DGAP 103. I think you'll appreciate, somebody had listed as mild facial dysmorphism. I'm not sure that any clinical geneticist would say that was mild, but you can see the dental overgrowth, the lipomas. This is just the histology of the lipomas. Here's the brachydactyly. You can see those enlarged interphalangeal joints, the bowing of the legs, and the sort of flared epiphyses. So we don't really have to refer to this patient by DGAP 103 because he has a Wikipedia page. And this is Brendan Adams, and he has a Wikipedia page because he's known as the tallest teenager on Earth. So Brendan was 7'5.5" at age 14, and he's now about I think it's 7'8", and his height was slowed by high-dose testosterone treatment starting at about age 14, 15. His physicians estimated that he would have been over 8 feet tall had that not been done. So here's a picture of Brendan going into the Seattle Children's Hospital when he was about 14 at that time, and you can see him with his mom and his half-brother. And I think you'll also appreciate lots of the things that we saw in the pictures of him as an 8-year-old. You can see the lipomas, you can see the facial dysmorphism, and certainly the incredible growth in height. And here's again the issue of the dental overgrowth. So at this point, Brendan had the previous year to this, he had 12 additional molars taken out. But he was in the hospital to have his spleen removed. And I think you can all appreciate that this is an enormously large spleen. And here again, the brachydactyly. So every once in a while, the genome gets smaller, and it turns out that knowing I forgot to point out on the ideograms, he has a pericentric inversion on chromosome 12. So disrupting a segment on the short arm at P1 1.2 and on the long arm at Q1 4.3. So this is a region that was near and dear to my heart because at 12q1 4.3 resides a gene known as HMGA2. HMGA2 is dysregulated in uterine fibroids and other mesenchymal tumors such as lipomas. So seeing that the clinical history included lipomas, we were able just to walk to the freezer, take out the cosmids that we'd been using to look at HMGA2 rearrangements, and determine that his inversion disrupted HMGA2, and it disrupted this gene between the 5' AT hook domains and the 3' acidic domain of this protein. So one of the things we need to do in this project is validate the phenotype and the association with the genotypic rearrangement. So let's take a look at DGAP 103's phenotype and what we know about HMGA2. So it turns out that HMGA2 truncation had already been observed in 2 mouse models with lipomas and overgrowth, and also to be rearranged in the synovia of patients with osteoarthritis. Subsequent to us publishing this case, it turned out from GWAS study that variants in HMGA2 influence human height. And I always like to kid my colleagues. It took us 1 patient to actually find a gene that was involved in human height. But the GWAS study here was in almost 19,000 or adults and other tall, short, case control studies. But HMGA2 is still the gene that variants in it singly explain the most of population variation in height, which is about 0.3%. But what about the brachydactyly? Well, one afternoon while I was editor of the American Journal of Human Genetics, this paper appeared on my desk from Stefan Munlo's lab, and it was entitled Deletion and Point Mutation of Parathyroid-like Hormone Caused Brachydactyly Type E. So I knew parathyroid hormone-like hormone because it was the gene that was closest on the other side of the breakpoint of this inversion. So it seemed very likely that that was the explanation. But we really never did any other type of experiments to confirm that. But it just seemed impossible that it wasn't due to some dysregulation of this gene. So in May of this year, there was a paper out of the Mundlos lab that I just remain incredibly excited about, and I hope if you haven't read this paper that you'll take a moment to do so. And this was also presented at the European Society of Human Genetics meeting in Glasgow this year. So it's entitled Disruptions of Topological Chromatin Domains Cause Pathogenic Rewiring of Gene Enhancer Interactions. So what are topological associated domains? These are megabase-sized genomic segments, and they partition the genome into regulatory units where there are frequent interactions within the unit, so sort of under the tent, but relatively rare interactions between different domains. They're very conserved among cell types and species, and they're considered to be crucial for spatiotemporal gene expression patterns. I just find this so satisfying because when there's order to our system, it leads to explanation. So the topological domains, or the TADs, are separated by these transcriptional boundary regions, which are called TBRs, or you may know them as insulators from classical work in Drosophila, and they block the interaction between these TADs. And disruption by structural rearrangements will cause rewiring of genomic regulators that can result in abnormal phenotypes. So I want you to think about not only structural rearrangements that are mediated by translocations and inversions, but also think about microdeletions and microduplications and how this might impact an understanding of what the phenotype is that's associated with these. So there's a really nice overview of how this works and with various rearrangements. The top part of this cartoon is the wild type situation. So you can see two TADs, A and B, located next to each other, and then this insulator element between them, and an enhancer in, is it TAD B? TAD A is actually impacting gene 1. And that enhancer is not interacting with the gene in the next door TAD. But if you have inversions, you can actually flip that enhancer into the other TAD, and it can act on a gene that it doesn't normally act on, and then not do its job with gene 1. And if you have a duplication, you can cause a different situation. It's a little hard for me to see this on the screen because it's tiny here. But if you think about the situation of a deletion, if you take out that insulator region, that enhancer might work on both of these genes. But if the deletion is totally within the TAD, there might not be a change on the expression. So the highlights of this paper are that disruptions of these TADs will lead to de novo enhancer-promoter interactions and misexpression. And that occurs when the TBR, the transcription boundary region, associated TAD boundary elements are disrupted. So structural variations that disrupt the TAD structures can cause malformation syndromes. And in this paper, they did a proof of principle experiment using CRISPR, and it's just beautiful experiment. And different phenotypes can result from one enhancer acting on different target genes. Seems very straightforward. So what about the situation with brachydactyly? So it turns out that when we map the breakpoint of that rearrangement, it's within a 1.32 megabase region that includes parathyroid hormone-like hormone. So it's located in that specifically in that transcriptional region, and it was at a distance of 806 kilobases telomeric to the breakpoint on the short arm. We had actually looked at it with respect to whether there might be some type of a fusion transcript from HMGA2, and there was no evidence of that. Beyond that, we can look at the haploinsufficiency index of the genes that are in that TAD. There is only one gene in that TAD that's predicted to sort of have an impact by gene dosage. And this comes from the data from Decipher. And you can see here that gene is parathyroid hormone-like hormone. So I feel that we now have a very reasonable interpretation of this phenotype of Brendan Adams. How about using this type of approach in prenatal diagnosis? So I'm going to describe for you our foray into this with 10 pregnancies. All of these were considered to be high risk for either advanced maternal age or some type of abnormal maternal screen or ultrasound. All 10 of them had balanced de novo rearrangements, and importantly, all of them had normal array CGH results. So we know from some elegant work of Dorothy Warburton in early 1990s that the risk of an untoward outcome from a diagnosis of a de novo balanced rearrangement in a prenatal setting is 6.1% for a translocation and 9.4% for an inversion. Of course, these babies were basically, or were largely assessed only in the newborn period, and it remains to be known whether these are underestimates or maybe overestimates based on the fact that any ones that had abnormal array CGH results probably were, at least presently, would not be in this group. That being said, I had a hallway conversation with Niels Tommerup 2 years ago where he told me that they had reassessed the outcomes in the Danish registry of individuals with balanced de novo rearrangements, and the risk was more on the order of 20% for some untoward outcome. So how do we actually do this prediction of phenotype, and where are we now moving? So we call this our convergent genomic evidence analysis approach, which the whole approach until relatively recently was what's here in the first bullet point. We looked at the candidate genes that were at the breakpoint or in the vicinity of the breakpoint for disruption or dysregulation, and reviewed the literature, looked at OMIM gene map and morbid map, the Decipher and DDG2P, everything we could get to try to find out whether there was a human pathology that was associated with the gene that was involved in the rearrangement. But now, since this summer, we're also looking at phenotypic associations relative to the TADs and TBRs, and we're going to go back and reinterpret, it's kind of scary, the cases that we already did. And I'm going to talk to you about those. And then we're also using the information from Decipher in terms of the haploinsufficiency indexes for the genes that are in the TAD. So the first case is a case that we published in the New England Journal of Medicine a couple of years back now. It was the first sort of whole genome sequence of a prenatal case, although of course we weren't doing that sequencing looking for single nucleotide changes. We were doing that looking for structural rearrangements. This was a 38-year-old with a history of infertility, and this was a second pregnancy following IVF. And she had normal first trimester screening. At about 19 weeks gestation, there were concerns about the heart, where there was tricuspid atresia and hypoplastic right ventricle. And she and her husband did not choose to have any invasive prenatal testing, because this was a very wanted pregnancy, and they were reassured by multiple visits to pediatric cardiologists and surgeons in Boston that with a few surgeries, their child would live and essentially a normal life. But at 27 weeks, the situation became more morbid with polyhydramnios and an undetected stomach. And then at 33 weeks, there were additional abnormalities, and due to the polyhydramnios, they began to have therapeutic amnioreductions, and from one of those amnioreductions, we received a sample for karyotyping. So this is the perfect test to get perfect patient to get your feet wet on. We're already 33 weeks out. It's not a question of whether this, the result that we might come about from sequencing would be used in making any decision about the pregnancy. Also at 35 weeks, there was noted an undescended testis. So the karyotype revealed an apparently balanced rearrangement between chromosomes 6 and 8, and there was a normal array CGH that was performed. What we learned from the sequence analysis is that, yes, okay, oh, sorry. That there was, the breakpoint was actually in Q1 2.2, and you'll see the common theme of the slides. I'm going to show you that if there was a change in the breakpoint, I've indicated it in green on the karyotype, and then any alteration in that in red, and you won't be surprised that we often change the assignment. I think that in defense of many cases that I signed out over the years, some of these cases are complex at the break point, and it's not simply a swap, so the banding pattern is a little confusing at that point. You just can't really line it up perfectly. But what you can also see is on those ideograms, you see the part of the shading of that chromosome that's in green. That's the precision from the cytogenetic analysis. And of course, with the sequencing, there's a single line drawn through there, which is exactly where the breakpoint occurred. In this particular case, there were two genes disrupted, LMBRD1 and CHD7. And the clinical diagnosis at birth was CHARGE syndrome, which was completely consistent with the clinical findings at birth and with the findings from the prenatal evaluation. But there had never been a part of the differential diagnosis, and this child expired at 10 days after delivery. So let's take a look at the TADs in that region. So it turns out that on chromosome 6, you can see where the breakpoint is with this blue line. It disrupts LMBRD1, and this is a gene involved in cobalamin metabolism, would be consistent with metabolic disease, but this would require both alleles to be involved in an autosomal recessive fashion. And then you can see that on chromosome 8, we have the disruption of CHD7. So in the convergent analysis here of this, there were no other genes that we worried about in terms of haploinsufficiency or imprinting that were associated with an abnormal phenotype other than CHD7, in which CHARGE syndrome is well established. So second case is also really was valuable for us because the mom herself is a scientist who clones mouse chromosome rearrangements in her professional life. And so when her child had an inversion on chromosome 8, she was a participant in the analysis. So she was a 41-year-old. This was her third pregnancy at 16 weeks. Again, normal first trimester screening, an unanticipated inversion, a paracentric inversion in chromosome 8. Not surprisingly, we reassigned the breakpoints, and one breakpoint was in a non-genic region, and the other disrupted a gene known as KHDRBS3. That gene is an RNA binding protein that's involved in the regulation of alternative splicing. We counseled her that there was no pathology associated with KHDRBS3, and they decided that we could find by the literature, and they decided to continue the pregnancy, and the child is doing well now, even must be about 8 months old now, so everything has gone very well for them. So you can imagine thinking about looking at the TADs on this one was a little scary for us, because she had really become, like I said, our partner in this analysis and our friend. So one thing we did do was do qRT-PCR looking at the expression of KHDRBS3, and perhaps not surprisingly, you can see that in age-matched amniotic fluid cells on the top, and also in age-matched cord bloods on the bottom, there was a decrease across the board in this gene. So the TAD analysis does not reveal that there's any other genes that are associated with an abnormal phenotype, other than, of course, the disruption in KHDRBS3, which we were not able to associate with any abnormal phenotype. So we gave a big sigh of relief over this, that perhaps, in fact, there won't be any surprises down the road for them. But of course, we always interpret these cases, or especially at this point where we didn't have this extra ability to look at our organization of our genome, as variants of unknown significance. Okay, this is the third case. This was a 44-year-old, first pregnancy, 12 weeks. She had a CVS. She had a normal first trimester screening, and we found a de novo 213 chromosome translocation. And again, revision of the breakpoints. In this case, there was one gene disrupted, RFC3, which is an accessory protein that's involved in DNA replication and is a clamp loader. Unfortunately, for us at that point, when we called to schedule an appointment for the couple to come in and talk to us about the sequencing analysis, and believe me, trying to get this done in an actual time frame, so we're trying to get it done under 2 weeks. It's an all hands on deck experiment, if you like, because of course this really is research. We found that she had terminated the pregnancy prior to coming in to talk to us about the sequencing results, and basically told us it was a multi-layered decision. So we were all kind of heartbroken, because we thought this case might have actually a chance to sort of be like the case with the previous case, in which we couldn't find any pathology associated with the disruption of that gene. And we did do qRT-PCR also, and could show that the gene that was disrupted showed less expression compared to age-matched CVS cells. When we looked at the convergent analysis with the TADs, we found that there was nothing in the chromosome with RFC3, but the breakpoints in the non-genic region involved with chromosome 13 are upstream to NBEA, so within that same TAD, which is a candidate haploinsufficient gene for autism. So I guess you could say we've had 3 cases that we really it was great to have these as our training ground, because this case, how would we have handled this information with her? I'm not sure exactly, but this would have been something that really would have been problematic in terms of sort of, again, flying the plane while building it. I mean, delivering this type of information. Okay, the next case was an IVF twin pregnancy, and when I got called on this, I was like, twins, please. Okay, so there was an abnormal maternal serum screen, and we determined that the twins were MZ by the SNP microarray, and because all these cases always come at Thanksgiving, it seems like in the United States. If you're going to have an emergency, it's on Thanksgiving in the cytogenetics lab. So because they were MZ, we were able to combine we decided to combine the DNA that was left over from the array CGH and do the make the library with that. These twins had a de novo pericentric inversion involving chromosome 6. And of course, again, we revised the breakpoints, and certainly at that time, I felt great. They're both in non-genic regions. This is the best diagnosis you can get from us at this time, and the babies were normal at birth. So it turns out we found another inversion that we weren't expecting, and it's a cryptic inversion, so you can see that little purple segment, and that turned out to be inherited from the dad. So it was somewhat comforting to us that we would find things that we weren't looking for or didn't expect by this analysis. We still have yet to actually determine whether the inversion is on the chromosome, on the derivative 6 chromosome. Was it inherited from the dad, and was the rearrangement, did this occur in the father? And by the way, most of these types of de novo translocations are paternally, from the paternal contribution. But in looking at the TAD analysis, that inversion is in the same TAD with the rearrangement. So we really are keen to know whether it is there and whether these types of sort of cryptic rearrangements in the genome might be the seeds of some destruction, that this is what actually mediates these other larger types of rearrangements. But at least in terms of these twins, there was no other thing that we needed to be concerned about based on the analysis. Each case is different. So this is a case of a 21-year-old who had a clinical finding. On the ultrasound, there was bilateral ventriculomegaly and partial agenesis of the corpus callosum. There were five chromosomes involved in a rearrangement, so this is a germline chromothripsis case. And you can see there was one translocation that was a 4-way with 3, 18, 5, and 7, and then another rearrangement involving chromosome 9 and 18. And again, they decided based on the clinical findings from the ultrasound and this picture that they would terminate the pregnancy. So the pressure was off of us time-wise to deliver this analysis, but we were able to solve all of these breakpoints. And of course, the TAD analysis is huge here in looking at all these places, but I can tell you that one of these breakpoints is in an area of regulation for sonic hedgehog. So there are various things here in this analysis that would be consistent with the complex brain phenotype that was seen. Just a couple more cases. Here's a 36-year-old G3P2 at 12 weeks who had abnormal fetal ultrasounds, a cystic hygroma and micronathia. And there was a balanced rearrangement of 6 and 17. Again, we changed the breakpoints. And you can see that in this case, both of these rearrangements were present in non-genic regions. Interestingly, we were able to, looking at the analysis, get suspicious about SOX9 because it was in the neighborhood. And we did qRT-PCR, and you could see that there was decreased levels of SOX9. And the fetal MRI and postnatal findings were actually suggestive of isolated Pierre Robin sequence, for which there are variants upstream of SOX9 that are responsible for this. So we did the TAD analysis and found that SOX9 is in the same TAD, 1.8 megabase TAD with the breakpoints. And with also these well-known cis-regulatory regions for Pierre-Robin sequence. So it seems to fit the clinical findings. Then another case of a 38-year-old who had an IVF pregnancy with abnormal first trimester screening, and this time there were 2 genes disrupted, and there was a bit of complexity in the rearrangement, and that there was an inversion also at the breakpoint. And that's actually not an uncommon finding either. So the case of the twins I showed you, that inversion wasn't right at the breakpoint, but we've seen a fair number of DGAP cases that actually have inversions associated with the rearrangement. And in this case, let's see, the 2 genes that were broken were really determined to be of unknown clinical significance. But this couple decided to terminate this pregnancy based on the fact that 2 genes were disrupted, which we interpreted again of unknown clinical significance, and the fact that there was complexity at one of the breakpoints. However, our TAD analysis would have suggested, of course, that was subsequent to this, because at that point we weren't doing that, or we weren't knowledgeable to do that. So there was no evidence of anything associated with an abnormal phenotype. So we, at least with our limited knowledge of this, we would have been a bit more optimistic about this case. And I think this is maybe the last one. It's a 21-year-old at 19.4 weeks with abnormal first trimester screening and fetal ultrasounds, very notable for growth restriction. And this was a 211 rearrangement, and the sequencing analysis revealed a really complex inversion that was involved in the chromosome 11 in addition to the translocation. And this disrupted on chromosome 2 a gene known as GFPT1, and didn't seem that that would be likely to be involved in the phenotype. But on chromosome 11, we were really in the area of Silver-Russell or Russell-Silver syndrome, and we're concerned about the possible hypomethylation of H19 IGF2 imprinting control region. The baby was actually, I think, born at, I don't remember the weeks, but it was a 1-pound baby, and there was severe intrauterine growth retardation and poor postnatal growth. So when we do the TAD analysis, we find that the breakpoints are located within the 600 kb TAD that includes IGF2. So seems like that might also be important. And we haven't been able to confirm this in a sample from the baby because it's still a pretty small baby, and we didn't have enough DNA to do, or any sample, additional sample to do the qRT-PCR, but we're hoping that that will be possible in the future. So this is just a summary of sort of the clinical outcome of these cases that we've looked at compared to the predicted clinical significance. And we're feeling that this is going to be a part of the way that we go forward in terms of interpreting structural rearrangement. So not only in terms of these types of cases, but also thinking, like I said, about microdeletions and microduplications. We've sort of focused our interpretation by looking at what's deleted inside that microdeletion. But maybe we better position that microdeletion with respect to where the topological domains are and start looking at genes that are in that neighborhood. And I'll just end quickly telling you a little bit about what has been a, I don't know, I want to say a baby of mine, which is to try to establish a nomenclature that allows us to present this information. And it looks sort of scary, but I can assure you the more you look at it, the more sense it makes. And I feel that this will be a future for us in terms of, again, making a nomenclature that we can do big time data sharing with people around the world and know exactly what the structural rearrangement is. And we've developed a a poor algorithm at this point to try to do this for the cytogenetics community. If you had the sequence across the breakpoint, you could stick it in this program, which we call Boston. And there's a reason why we did this, because there were 4 notable conferences in cytogenetics. There was Denver, there was London, there was Chicago, and Paris. So when I said, oh, we have to call this Boston, because there's not going to be another conference to sit down and straighten out the cytogenetic nomenclature. And my very talented postdoc, Zara Ortalu, came in and said, oh, well, it stands for Blast Output Sequence Tool of Nomenclature. So there you go. It was meant to be. Okay, so just to summarize, I think the evaluation of genes disrupted or potentially dysregulated at breakpoints requires nucleotide level precision for clinical interpretation. And we'll use the TAD and HI analysis with convergent genomic evidence to more accurately assess potential clinical significance. We can incorporate expression analysis, qRT-PCR studies, to try to expand our assessment of the potential positional effects. Nucleotide level resolution of balanced chromosome rearrangements is possible in an actionable time frame for prenatal diagnosis. And the rearrangements may be complex or simple in the setting of normal array CGH results. And I'd just like to say thank you especially to all the families who trusted us to provide information for them. So with balanced rearrangements and with abnormal phenotypes, and the honor roll of all the healthcare providers who have participated in this journey with us. And here's the current team after we had just learned that our renewal was likely to be funded. So we were all pretty happy that day. And there's just an amazing group here, including this young woman and at the end of the second row, who's my genetic counselor who trained in Newcastle. And she told me the other day that her husband's looking at jobs to come back, and I was like heartbroken. Okay, so I said, okay, you have to find me another British genetic counselor to take your place, because I think American families are just so drawn in by that British accent that she's very successful in enrolling them. And then I'll just close with actually some words from T.S. Eliot, which Eric Lander used at the end of the Nature article on sequencing, because I feel this is so true also about how I am seeing this future in interpreting structural rearrangements in cytogenetics. So we shall not cease from exploration, and the end of all exploring will be to arrive where we started and know the place for the first time. So this is just, for me, becoming a very satisfying way to go forward in cytogenetics. So thank you very much.